The overall goal of these procedures is to demonstrate how to precisely transfer cells or DNA into early post-implantation mouse embryos.
These methods can help answer key questions in developmental biology, such as testing the in vivo potential of in vitro cultured cells and functions of certain genes in specific embryonic stages. The main advantages of these techniques are that they do not require any specialized equipment and render experimental manipulations of early post-implantation mouse embryo possible.
After sacrificing a pregnant female on day E7.5 or E8.5, according to the TEX protocol, use scissors and forceps to isolate the uterus and place it in a 30mm dish filled with M2 medium. With 2 pairs of fine forceps, carefully tear apart the myometrium. Then, peel away the deciduas, being careful not to puncture the extraembryonic cavities. Remove the Reichert's membrane by using forceps to pinch it and slowly separate it from the embryo. Then, under a stereodissecting microscope, check the embryos to ensure that the yolk sac, amnion, and ectoplacental cone are intact. With a pipette, transfer the embryos to a clean dish of M2 and place on a 30mm plastic petri dish lid on ice or an ice platform to partially chill the embryos. Prepare culture medium and culture embryos according to the TEX protocol.